Everyone, I am Dr. M. Parimala, Assistant Professor, PG and Research Department of Microbiology, St. Joseph's College of Arts and Science, Autonomous Cuddalore, Tamil Nadu. I am going to talk about affinity chromatography. Now let's see what is affinity chromatography. Affinity chromatography is a separation technique in which a mobile phase carrying a mixture is caused to move in contact with a selectively absorbent stationary phase. This chromatographic technique involves the usage of a stationary phase as well as a mobile phase. In the mobile phase, we have to add the mixture in which the analyte has to be separated. Affinity chromatography is a type of liquid chromatography for the separation, purification, as well as the specific analysis of the sample components. It utilizes the reversible biological interaction or molecular recognition called affinity, since it is called as affinity chromatography, which refers to the attracting force that extend in different degrees between the atoms which cause them to remain in combination. The example for this chromatographic technique is enzyme with an inhibitor, antigen with an antibody, etc. This technique was discovered by Pedro Kutrakis and Mayer Wilczek. In this diagrammatic representation, it clearly indicates how an analyte can be separated from a mixture through the mobile phase when it is passed through the stationary phase. The stationary phase will be a supporting material or a matrix in which we have to bind a ligand. The ligand is the specific substrate to which the analyte will bind. Then after we have to add the mobile phase along with the mixture that contains the analyte to be separated. It will be passed through the stationary phase, that is the supporting material, and in the stationary phase there is the specific component called ligand. This will be having specific binding site for the analyte to be separated. So through this affinity, the molecule to be eluted that will move through the mobile phase and it will get bind to the ligand. Then it will be gets eluted. The analyte will be eluted, and once again if you wash with buffer, the stationary phase will be equilibrated once again. Now let's see the principles of this affinity chromatography. The stationary phase consists of the supporting medium on which the substrate, that is the ligand, is bound covalently in such a way that the reactive groups that are essential for binding of the target molecules are exposed. So these ligands are found to be very much specific. So in this ligand only the mixture containing the analyte which pass through the mobile phase will go and bind. As the crude mixture of the substance will pass through this chromatographic column, the substances with binding site for the immobilized substrate that bind to the stationary phase using this ligand, while all other substances is eluted in the void volume of the column. Once the other substances are eluted, the bound target molecule, that is the analyte which has to be separated, can be eluted by using some methods which includes the using of a competing ligand in the mobile phase, or changing the pH or ionic strength or polarity conditions. So by doing these changes we can elute the analyte. Now we will see the important components that is actually involved in this affinity chromatography. The first and foremost important thing is the matrix. The matrix is the inert support to which a ligand can be directly or indirectly coupled. So this is the stationary phase material that will be packed. In order for the matrix to be effective, it must have certain characters. The matrix should be chemically and physically inert. That is, it should not be reactive with either the analyte to be separated. It must be insoluble in solvents and buffers employed in this process. It must be chemically as well as mechanically stable. It must be easily coupled to a ligand or spacer arm onto which this ligand can be attached. It must exhibit good flow properties and have a relatively large surface area for attachment. The most powerful matrix materials used for this process are agarose and polyacrylamide. So this matrix, it is a very important component in the stationary phase, to which the ligand or the spacer arm will be bound so that the analyte to be separated while passing through the mobile phase, it will get specifically bound to the ligand. And next important component that is used for this chromatographic technique is the spacer arm. It is used to improve the binding between the ligand and the target molecule by overcoming any effect of steric hindrance. So this spacer arm will improve the binding, that is in between the ligand and the analyte which has to be separated. The third important component is the ligand. This molecule that binds reversibly to a specific target molecule. The ligand can be selected only after the nature of the macromolecule to be isolated is known. So the selection of this ligand molecule is found to be very specific. When a hormone receptor protein is to be purified by affinity chromatography, the hormone itself is an ideal candidate for this ligand. And also for the isolation of an antibody, the antigen or the hapten can be used as the ligand. If any enzyme to be purified, a substrate analog, inhibitor, cofactor or effector may be used as the immobilized ligand. So the ligand is exclusively used for binding with the analyte, that is the component from a mixture has to be separated. And now let's see the steps involved in affinity chromatography. First thing is, affinity medium is equilibrated in binding buffer. This is the first step. Then the sample has to be applied under conditions that favor specific binding of the target molecules to the complementary binding site. So this is the next thing that we have to do. The sample will be applied at a condition so that it can bind specifically with the ligand. The target substances bind specifically but reversibly to the ligand, and the unbound materials will be washed through the column. So this will happen at the initial phase. So the specific target, that is the analyte which has to be separated, will be bound to the ligand. The remaining unbound molecules will be washed out through the column. Then the elution is performed specifically using a competitive ligand or non-specifically. You can change the pH or ionic strength or polarity. So you can, after we have to make this, the separation of the unbound molecules, we have to make such changes. So this is optional, and you can do this by either changing the pH or ionic strength. Likewise, you can alter. Then this target protein will be collected in the purified, that is the concentrated form. So this is one of the best example for how this molecule can be separated using this affinity chromatography. Affinity medium will be once again re-equilibrated with the binding buffer. And now we will see the three major steps involved in this process. First one, preparation of column. The important component, that is the column, is loaded with a solid support such as saccharose, agarose and cellulose, etc. The ligand is selected accordingly to the desired isolate. That is, it depends on the analyte. We have to select the ligand. And the spacer arm is attached in between the ligand and the solid support. So the stationary phase preparation is a prerequisite. So this will be carried out using this first step. Then the second step, loading of sample solution containing a mixture of substances that is poured into the elution column and it is allowed to run at the controlled rate. So this will be the addition of the mixture, that is the sample in which the analyte is present has to be separated, that will be added and it will be eluted. Then the third important step is the elution of this ligand molecule complex. The target substances is recovered by changing the conditions to favor the elution of the bound molecules. So after binding, the elution process can be carried out successfully by making such modifications. And now let's see the applications of affinity chromatography. So what are the major applications of this affinity chromatography? Affinity chromatography is one of the most useful methods for the separation and purification of specific products. So many biological products can be separated using this technique. So this is found to be a complementary one like antigen antibody reaction or enzyme inhibitor complex, etc. So it is found to be very specific. So based upon the ligand, that is based upon the product or the analyte to be separated, we have to select a suitable ligand and the separation process can be carried out successfully. Then it is essentially a sample purification technique used primarily for biological samples such as proteins. So whenever we want to separate the and purify the protein mixture, you can carry out this process. So you can bring about the purification of this protein molecules, etc. Then its major application includes first one, separation of mixture of compounds. If there is the mixture of compound that will be present, we can separate out the desired one, that is the analyte. Next one, removal of impurities or in purification process. So while purification, we have to remove the impurities so that we can get the purified product of either a protein or any other substances. So you can carry out this very easily using this affinity chromatography. Next one, it is used in enzyme assay. So since the enzymes are protein in nature, this will be very much useful for enzyme assays too. Then it is used for the detection of the substrates. This method is very much useful for the detection of the various substrates and also the investigation of binding sites of enzymes. So for enzyme assays, the binding site is found to be a major thing that is very essential for this enzyme activity. So we can find out that we can investigate the binding sites for different enzymes. Next one, it is also used in the in vitro antigen antibody reactions. So the in vitro under laboratory condition, we can find out the antigen antibody reactions. So the antigen antibody reaction, it is found to be very specific. So it will be found out under laboratory conditions. So this will be very much useful to study these reactions under laboratory condition. Then the next one is detection of single nucleotide polymorphism and mutations in nucleic acids. So this separation technique is also very much useful for studying this mutation studies in nucleic acids. Finally, we can study the advantages of this affinity chromatography. Regarding this advantages, it is found to be highly specific, as I mentioned, like antigen antibody reaction or enzyme substrate or enzyme inhibitor complex. So the specificity can be very much useful. The target molecules can be obtained in the pure state. Then this purification step is found to be very simple. Then the matrix what we are using can be reused rapidly, so we can reuse them. Then the matrix is a solid, can be easily washed and dried. So we can reuse it again and again, and we can get the purified product, and also it gives the high yield. The yield is found to be very high, and this affinity chromatography can be also used to remove specific contaminants such as proteases, etc. And now let's see the limitations of affinity chromatography. It is a time consuming method, then more amount of solvents are required which may be expensive, then it needs intense labor, non-specific adsorption cannot be totally eliminated, it can only be minimized, then limited availability and high cost of immobilized ligands. Cost base is found to be high. Then proteins get denatured if the pH is not properly adjusted. So these are the some of the limitations of this affinity chromatography. Overall, this affinity chromatography, it is very much useful for the separation of the biological compounds. Thank you.